this next chapter, which is on hormonal regulation. So we're going to go into greater detail now using some of what, building upon this last chapter since we ended with looking at some of the hormones, neurotransmitters that the body uses. In this chapter, we're going to go into greater detail. Okay, so what are hormones? By the way, if you've forgotten some of this from A&P, the Khan Academy on YouTube does have like a little 9-minute primer that I suggest that you can go over, because it goes over like some of the more detail of the anatomy than what we'll cover today. But what are hormones?
Signals, messages.
Signaling messengers. Okay, that's correct. And so, what are some of the examples of hormones? Testosterone. We all have testosterone. Some of us have more than others, hopefully. What's another? Estrogen. I see where your mind's going. Okay, progesterone, prolactin, that's follicle stimulating hormone. What about some that are non-sexual hormones? Dopamine, insulin, glucagon, epinephrine, things like that. So, wow. And so, hormones in general, they're going to integrate and coordinate metabolic activities. And essentially, in mammals, even in non-mammals too, the hormones, every metabolic process is dictated by one or more hormones. Okay, it's a coordinated activity. It's a way to optimize what your body needs and uses. So that way, it's not, if everything is under control, then it's not wasting materials. Okay, and so virtually every process, in those of us that are complex organisms at least, is regulated by one or more hormones, whether it's sexual differentiation, which you guys really honed in on this last, when I asked you for examples, to electrolyte levels, blood pressure, on and on and on. Touch, every process that you can think of would be essentially regulated by at least one or more hormone. Okay? And in mammals, it's achieved through what's called the neuroendocrine system. Right? So, what are the two parts for neuroendocrine? Oh, whoops. This is the wrong one. It's not what I meant to do. Neuro and endocrine. Well, neuro, what does the neuro mean?
Brain.
Is it only the brain? Right, we're talking about neuronal. The neuronal system versus the endocrine system. Okay, what is the big difference between the two? What makes the neuronal system versus the endocrine system? Not necessarily.
Would it be like fast versus slow acting?
No. Why? Because which one are you saying is fast?
I'd say the
So there are no fast endocrine systems?
No, there's the character.
So, what is the big difference? If you're trying to explain the difference between the two to someone who, in layman's terms, there is something that separates these two.
The neuronals more electrical?
No, not necessarily. Technically, even the movement, electrical just means movement of electrons, right? So lots of things can be electrical.
Endocrine be like outside the nervous system? I don't know.
Wait, what did you say? No, but you're getting closer. No.
Is it location? Is it receptor? Or is it the secretion part?
It's distance. Distance. And so, just typically, the neuronal, what kind of distance is a neuronal? We're talking about synapse, right? And so here we're talking usually fractions of a micrometer. So fractions of, very, very tiny distances. That's why you're thinking, electrical impulse, the nerves going down, telling your muscles to move, but each signal just travels such tiny distances. Whereas the endocrine system can really range. Usually, we're talking, even if it's in the autocrine part, it can still go within that tissue larger distances than a fraction of a micrometer. Okay. In fact, there are some that the target is a meter or more away. All right, so what's an example of one that travels a long distance?
Well, it would be like a neuronal hormone, like maybe like something from the pineal gland that would travel like all the way down.
If it's going through the nerves, though, then it's only going from little tiny spot to little tiny spot because of the fact that it's... So what's one that doesn't? Well, in general, what happens with an endocrine hormone? Now, someone did say this earlier.
Secretion.
You secrete it many times, or almost always, and so what happens then? It travels through the blood. So what's an example of one of those? Insulin, right? So insulin's made where? Pancreas, and what's one of the tissues that gets affected by insulin? Your muscles, your liver. We're talking about large. Some of these can go large, very, very large distances. Whoops. There we go. Okay, so just in general, the main difference has to do with the distances traveled by the chemical messenger. Once again, as I mentioned, neuronal, we're talking about just fractions of a micrometer, whereas the endocrine can be very, very large distances. You can also kind of think of it almost like the neuronal systems, like your telephone line or something, where it's just traveling, even as a fiber optic nerve. Well, this is a nerve, but fiber optic signal, whereas the endocrine would be more like the general Wi-Fi. It goes all over the place. Okay. So, how do these hormones affect cells? Okay. What must all of these have if they're secreted, whether it's an electrical signal or if it's one that's secreted in the bloodstream? What must they have? Well, they have yes, they have an origin, like an origin, good old origin story. They have a receptor, so we're talking about receptors. That's one of the possibilities. That is their target. Whether it's like acetylcholine or GABA that we've talked about in the past, or if it's insulin with the insulin receptor. And what about those receptors? What are some examples? Like, where can they be located?
Transmembrane.
So transmembrane, all right, so where would the receptor part of the transmembrane be? For insulin, for example.
Extracellular.
It's extracellular, so that's one possibility, is extracellular. Which this now has to try to reconnect because the endocrine system of the university is on the fritz, i.e. Wi-Fi. Oh, come on now. So, extracellular, I was attempting to write that out. I was trying to write out extracellular whenever the Wi-Fi went down. So hopefully it will now not die again.
Yeah, the Wi-Fi was getting in trouble too. I was over there.
So we had extracellular. I don't know. So that was one way. I'm just going to put receptors, extracellular. What's another place for the receptor to be? instead of extracellular, intracellular. Or you can then think about, there are different parts of the intracellular, and so we're going to just say cytosolic. We actually talked about one of those examples last semester, very, very end of last semester. Remember, like with guanylyl cyclase, it could be an intracellular nitric oxide signal. What else inside the cell can have a receptor? Nucleus, and so there's nuclear. Right? Now, once it binds to a receptor, regardless, what has to happen? You get a response. Okay? So I'm not going to do that. So there are lots of different responses. So we've seen some of these responses already. Okay? So what were some of the possible responses that we've talked about in the past? So we have those secondary messengers. Exactly. So we have the secondary messengers. So that's like the cyclic AMP, the inositol signaling pathways, things like that. Okay, and that was due to allosteric regulation, was the little buzzword that they like to talk about there. We had the binding on the outside, caused the conformational change, and then had the secondary messenger on the inside. What was another one instead of one that had the secondary messengers through allosteric regulation? Like the GC, those are the GPCRs, G protein coupled receptors.
It like changes the conformation of the cytoskeleton.
Right. Mm-hmm. Those were the adhesion ones, like the integrins. So the integrins, where you could cause change for the cytoskeleton. It could also change the morphological features on the neighboring cells and things like that. Does anyone remember what some of the others were? There were 6. We had the ion ones, the ion gates, or the membrane potentials, if you wanted to think of it that way. There were 3 more. What one was the autophosphorylation one? It's very specific. In fact, it had one of the amino acids in the name. RTKs, the receptor tyrosine kinases. Where they phosphorylate each other, you scratch my back, I scratch yours, kind of thing. That was insulin, was an example of that one. So we have 4 of the 6. I always have to now think about which ones. Oh, yeah, we're missing one that I've already mentioned before. So we had the nuclear ones, which in particular we're talking the steroids or steroid-like. So those were in the nuclear. And then the other one that I'd already mentioned before, which was the receptor guanylyl cyclases, which could be either similar to the GPCRs or intracellular. And so I'm running out of room here to write, but those were the receptor guanylyl cyclases. Okay. So that's one way that you could discuss, the hormones. Then there's actually another way to differentiate the types of hormones too. And that has to do with their solubility. Oh, come on. I was going to say it's starting to slow down again. So these were just the 6 intracellular consequences. But we can also talk about the solubility. So we have those that are water soluble versus those that are not water soluble, which in vivo we usually call those, instead of being water soluble, they're called the lipid soluble or fat soluble hormones. Just in general, do you suppose that a water soluble is going to be able to pass through the membrane, or do they typically bind extracellularly? What's an example of one of the water-soluble ones? Where would you find a water-soluble? It's in the blood. Okay, so is it going to typically pass through the membrane, or is it going to bind extracellularly? Extracellularly, for example, insulin is a water-soluble one. Okay, and the reason why they're going to bind, why would they bind? I'm not going to give you that answer. Rather than just passing through the membrane. They'd have to what? Right, they're going to be polar since they're water-soluble, so they're not going to be able to pass through the hydrophobic membrane, usually. They'd have to have some type of help to get across that in general. So that's why many of them, they tend to be extracellular receptors. Many times, these tend to be the amine hormones. We've talked about, like, in some of our classes, or even last lecture we alluded to some of these being called like catecholamines and things like that. So the amine hormones tend to be the water-soluble ones. So what were some examples of the water-soluble ones? I already give you one. What was the example that I gave you? Insulin. What's another example that would be water-soluble? Epinephrine. Adrenaline, noradrenaline, things like that. Acetylcholine. Okay, so, all right. And then what kind of response occurs whenever these bind extracellularly? They're going to have the cascade to where they bind extracellularly. There's some type of conformational response that causes the intracellular, response to the conformational change, I should say. So that's really the review from last semester, the very last chapter of last semester. So on the other end of the spectrum, we have the steroid or steroid-like hormones. So these are lipophilic. So if we just wanted to think about their relationship with, the membrane, are they going to be able to pass through the membrane or typically not be able to pass through the membrane? They typically pass through the membrane. It's the reason why last semester we talked about how the steroid ones tend to have their receptors even in the nucleus where they pass all the way through. In fact, my next question had to do where would the receptors many times are, and that's because oftentimes they are in the nucleus. Okay. If we compared the nuclear responses, for example, versus those that are the water-soluble hormones, what kind of response are we usually talking about here? They usually affect gene expression, transcription, things like that. So if they're going across into the nucleus, then we're talking about some type of transcriptional control instead of using the secondary messenger oftentimes. Okay, and so just a little cartoon picture depicting that to show where we have the peptide or the amine water-soluble hormones. It binds to the receptor. There's the conformational change or something. It modifies or activates a secondary messenger, which can go on and do a whole host of things in that cascade. Whereas the steroid or steroid-like hormones typically just go on through and they have an intracellular receptor or a nuclear receptor that will elicit the change at the transcriptional level, whether it's an activation or inhibition of specific genes. Okay, so assuming that if we look here at the, come on now, did it? I don't know why Wi-Fi is so slow today. So here in this, and it's okay. So there at the secondary messenger level, it can affect transcriptional control. Right? There are transcriptional effects whenever we talked about insulin in the past. Remember, it was a bifurcated system to where there were some that, if you went through all of the MAP kinase pathways, when they had the MAP kinase, MAP kinase, and so on and so forth, it did affect transcriptional control. However, we also can have some that go on and directly affect pre-existing enzymes. That's like the phosphorylation, where we had like pKa. And so then it can have a quicker effect because you don't have to wait for the proteins to be made, to be synthesized. They already exist. They're there. And so they just have to be turned on or turned off or upregulated or downregulated for those that had multiple allosteric controls, where instead of being like a light switch, maybe it was, causing a modified allosteric reaction. And so then we have the three methods of delivery. I forgot to have that hidden, but we have the endocrine, paracrine, and autocrine. So you can classify them accordingly. Autocrine is the one there aren't very many examples of that. That's where the cell that releases them contains the receptors. So it gets affected by itself, okay? Whereas endocrine is the one that gets released in the system, and the target cells or the receptors could be on tissues that are great distances away. So what really makes the difference between the paracrine and the endocrine?
How close they are.
How close?
Brain cells.
How close what is?
The cells that have that receptor.
The cells that have the receptor. Okay, it still gets released into the extracellular space. It still can be released into the bloodstream. It's just that the highest concentration of those receptors are on neighboring cells. So it's like a regional one. Okay. Then we have another way classification of hormones, which I've actually already been using this sometimes, like for example catecholamines, like what those are, the peptide hormones, things like that, where we classify them according to their origin or their structures. And so we can classify them according to their method of delivery. You can classify them according to their water solubility or lipid solubility. And once again, the Wi-Fi has gone out. No, I don't know, maybe there's a big drain. And so, like, there was, I stopped what was really sad was the person who's there leading them around didn't know where A211 was. She's like, I see A210, I see A212, and I don't know where A211 is. I said, it's that door right there. They were outside our door whenever I came in. I was like, oh, that's really, really sad. I'm hoping that person is a freshman and that they're not a higher level current student of SAU to not be in this part of the building at all, or not be able to use the logic behind. I would say, don't go to organic chemistry because you'd really struggle. All right. So I do expect you to know these classes of hormones. Some of these, I didn't hide just because of the fact is we don't go into a lot of detail with them in this chapter, and also you may not have had a lot of experience with them so far, and that's those that are the thyroid hormones and the nitric oxide hormones, because nitric oxide is nitric oxide. It comes from arginine and nitric oxide synthase. And we have talked about how that it's a second messenger with the receptor guanylyl cyclases. whereas the thyroid comes from tyrosine, but obviously it's going to be in the thyroid tissue, so we're not going to go into more detail with those. But we're going to start with the peptide hormones. I do expect you to know examples. Example, insulin and glucagon is those are like the classic textbook examples that are given for peptide hormones. So their synthetic pathway, since they're a peptide, obviously they're translated. They come from translation of genes. But how else are they synthesized? I mean, once that gene has been translated, what happens to it then, if you think about insulin in particular?
Change from the pro.
It changes not even from the pro. What's the very first form? Pre-pro. And so, but yes, they are cleaved, they are modified, and that's how they have a precursor form that gets, proteolytically processed. It's cleaved, gets cut. And so, in the cartoon, if you wanted to see it, I do want you to know insulin, how it's synthesized. Obviously, it's, pancreas and the beta islet cells of the pancreas. The original transcription results in the translation of the pre-pro insulin. They call them, even though it's a single peptide, it's called the A, B, and C regions. Pardon?
Why does it go like A, B, C instead of like around? Do you see what I'm saying? It goes A, C, and then B.
We're going to talk about that, just in a moment. Because of their location in the final product, so to speak. Because, if we were doing it based off of the normal labeling in peptides, you always start with the amino terminus, and end up with the C, the carboxyl terminus. And so it goes BCA then, but which doesn't make sense. First, the signal sequence gets cleaved off. And we actually will talk in greater detail about how that happens and the implications thereof. But then we have disulfides that are very important. But if you notice, the C peptide chain gets cleaved and it's released because it's not held in place by any disulfide bonds. And so it's the disulfide bonds then that hold the A chain and the B chain, the A peptide and the B peptide together. But they are not contiguous. They are only held together covalently through the use of disulfide bonds. It's the signal sequence that actually lets it know that it should be packaged into the secretory vesicles. And then, like I said, we actually go into much greater detail later on. The proinsulin is the way it's stored until the glucose level in the blood gets up, and we'll talk about the receptor that controls the glucose receptor that's involved here. And then a specific protease comes in, cleaves off the C-peptide to make the active insulin. But we'll talk about its regulatory pathway later on. Also, a single peptide can actually make many, many hormones. This is just an example. Please don't memorize all of the functions. You don't need to know what the POMC gene does. This just shows you that based off of where they are cleaved post-transcriptionally, then you can make multiple hormones from it. And so the proteases recognize where to cut once they see a specific signal. So, for example, consecutive, positively charged amino acids like lysine and arginine. So that's why if you had a mutation where the lysine or the arginine was mutated to anything that wasn't positively charged, you could get erroneous processing. Now, some of these we will talk a little bit more about, like ACTH and what it does, but we won't talk about, the enkephalin and all of that. Usually, the peptide hormones are in these storage vesicles, and they can be in really, really high concentrations where they're practically crystalline. Which, even though it wasn't a hormone, we already saw. Well, it wasn't a hormone directly. We already saw an example of that in that surgery when the person had the cholesterol ester cyst. Remember, they were so highly concentrated cholesterol that essentially it was crystalline. You can see the little crystals of it. Similar things happen here with the pro insulin, or the pro-hormones, like proinsulin and the pro-glucagon, things like that. And then the vesicle will merge through exocytosis. The capillaries near these glands are fenestrated, which means that what does fenestrated mean? They're holy. They have holes. So that's why they're permeable to the peptides. Next step are the catecholamines. They come from tyrosine. So epinephrine, which is adrenaline, norepinephrine or noradrenaline, things like that. And we actually talked a lot about epinephrine and its secondary messenger system. Okay. Now, we're going to cover a lot more about this later on and how that they're made. I do want you to know how they're made, know their functions. There are so many diseases associated with these that you really should know. They are made by the adrenal glands. Does anyone know where's the adrenal glands? On the kidneys. Do you know what adrenal means? What?
It's a pyramid?
No. It's adjoining or adjacent to the renal gland, which the renal gland is the kidney. Even though it doesn't do the same thing as the kidney. It literally means that it's next to the kidney. And so it doesn't do the filtration of the blood or anything like that. Okay. They are very similar in the way that they're stored and released like the peptide hormones, and so I've given some examples here of epinephrine and norepinephrine and things like that. But once again, we'll go into these later on, and I didn't check the most recent edition, but in the next to last edition, it was table 23.6, so chances are it's still the same table in the last edition. Whoops. Now we have the eicosanoids, which the example here are the prostaglandins, like PGE1. And they come from arachidonic acid, arachidonate in vivo, which I don't expect you to know the structure of it, except you should know that it's the one that's the PUFA that has 20, so it's the largest one that we typically go over. So these are the prostaglandins and thromboxanes and leukotrienes. They are not stored in advance, unlike the other two that we've talked about so far. So they are paracrine hormones. So you should know how they differ in their synthesis and storage than the others. Because these are synthesized from arachidonic acid. And we get arachidonic acid from where? Can we make arachidonic acid ourselves? It's going to look a little asterisk. Yes and no. No, we can't make it directly like de novo synthesis. However, because it's not an essential fatty acid because of the fact that its precursor we can get from plants. And then we can, modify a portion of it later on. But no, it's one of the reasons why you have to have your essential fatty acids that we talked about in previous chapters. And it is paracrine, so they typically only work in the region that they are released from. The next one are the steroids. So what's an example of a steroid hormone? What? Testosterone. Estrogen. All your sex hormones. Wait, what's the precursor for it?
Cholesterol.
It's cholesterol. Yep. These come from cholesterol. And we won't go into a lot of detail with respect to that. We have vitamin D type hormones, and the example for that one is 125 dihydroxycholecalciferol. And its synthetic pathway, where does it come from? It does not come from the sun. It's not like, imbibe me with cholesterol, raw. You know, God of the sun from Egypt. Faith integration is not our faith. Its synthesis can be activated from its precursor in order to make vitamin D, or the useful form of vitamin D. But what's the precursor? Does anyone know?
Probably should.
Yeah, you probably should. It's also cholesterol.
So, when people say you get vitamin D from the sun, it's that you're activating.
You get the active form, the useful form is activated due to UV light.
It's activated.
There's the relatively inactive form
Of vitamin D.
of vitamin D, which is the kind that you usually get in your diet. That's why even if there are people that can get a lot of the vitamin D, but it's the inactive vitamin D, even from like the supplements and things, you still are required to get UV light, or otherwise you will have vitamin D deficiency, because the fact is it's not useful. Right, it's not in its active form. It's not activated until you get
So
UV
the light
light.
activates it directly or activates an enzyme activate?
That one I'm not really for sure. I don't know a lot about if it's the enzyme that gets activated by the UV light, or if it's UV light that activates it in and of itself. If I had to guess, I would probably say it's probably enzymatic.
So we can't
So.
live in our basement and just study.
Pardon?
So we can't live in our basements and just study.
No. In fact, there was this like notorious picture that was done by National Geographic. Every year I always think, I've got to try to find it. Like years and years and years and years ago. Because where do they have vitamin D problems? What parts of the world have vitamin D problems? Sweden. What about here in the United States?
Like
Canada.
Canada's not part of the United States. Even though, sometimes we think of them as being a 51st state. Like Alaska, Washington State, Michigan is notorious for vitamin D deficiency. And so, because they don't get enough sunlight, okay? So what do they? But there's this notorious thing in Russia. I remember seeing this picture. They take, of course, it's very similar climatically to Alaska and Michigan and states like that. So they don't get enough in the wintertime. They don't get enough direct sunlight, and so there's this picture of all these kindergartners in like their public school system that are essentially wearing like little diapers or bathing suits and sunglasses, and they're standing against this wall in a big room where they're just being inundated by sunlight, or by artificial light. And so that's also a little factoid that came out. They made this big. Not that I'm pushing one political party or another, but there was this big. Back in 2008, there was this big to-do about the fact that the governor's mansion in Alaska for Sarah Palin had, tanning bed, how she had her own personal tanning bed. But in reality, it's really common in Alaska for people to have their personal tanning beds. It has more tanning beds per capita than any other state because the fact that they suffer from vitamin D deficiency. Just like in Michigan, there in Ann Arbor and Detroit, they had tanning salons on almost every street corner. There are so many per capita because you're not required, but so So whenever I first moved to Michigan, the first year was fine. My brother moved in with me. He was from the Mid-South. My brother was a model at the time, and he started breaking out in these horrible, horrible rashes. And in fact, it left scars on him and everything. So we went to the doctor, and the doctor looked at him and said, "I can tell immediately you grew up in the South." And he's like, "Yes." And she said, "It's because it's common here where after about a year, your body's not used to the lack of UV light. And so you have vitamin D deficiency, and it's causing skin issues. And so you're required to go and spend at least 15 minutes per week in a tanning bed." And so it's very, very common there to have tanning salons. Also, based off of your skin color, it does matter. So who needs more sunlight? Like what are some of the ethnic groups that need to have more sunlight than others? Pardon? It's African Americans. The darker your skin tone, the more UV light you need to activate your vitamin D. So if you're very, very pale, if you're one of these people who, go out and you burn immediately, you actually don't need a lot of vitamin D. It's, you just have some exposed skin going from building to building and you're probably fine. However, there are some studies that say that some really darkly pigmented people, because of the melanin in your skin, you actually need melanin, not melatonin. That's from neurochemistry that we had this discussion. Melanin in your skin that you can need sometimes as much as 20 to 30 minutes of sunlight per day. Okay. But that does not give you, I'm not giving you the permission to go down here to the tanning bed and turn yourself orange. So that's completely different.
That doesn't occur by bed. That's the chemicals that aren't good for you anyways.
Right, but the tanning bed itself is too highly concentrated of UV light for the vast majority of people. You usually only need no more than just a few minutes per day for anyone. And so, because of the fact is that you literally, it does, as Leon would tell you, it does highly increase and I should point this out, it's because of his capstone topic, not him personally. But it does greatly increase your chances of melanomas and other types of skin cancers. I had a friend who died at a young age. She was only about 50, I think, who died from skin cancer because she went to the tanning bed too often.
So does it matter if this is direct sunlight or indirect? Like if we're like in here and there's sunlight.
It would matter for in here because especially in Florida and southern states, a lot of times they put in filters in the glass that help filter out UV lights. So that way we don't get too much. Same thing. Now your eyes, you do need to protect, and so that's why you do want to get the polarized glasses that have both UVA and UVB. Because your eyes are different. You're not making vitamin D in your eyes, so you want to protect them.
This girl would go to the tanning bed twice a day.
That's really sad.
Twice a day. Well, she has that really nice glow right now. She hasn't got to the white spots and
Right, it
skin
will
cancer
age.
stage.
Well, it'll just makes your skin leather. It does harden the skin. You have to take a lot more moisture
But
in order
you're not
to
even
keep it up.
allowed to do that. She goes to 2 different ones because they won't let her do it.
Right.
But do you know how expensive that is when you can literally walk outside for free? She's like, that takes too long.
Yeah, and the reason why it takes too long is because when you're in the tanning bed, it's highly concentrated UV light. So that's not to say that just because you go to a tanning bed once in a while, that you're going to hell. Faith integration there, but that's not true either. So you will. Oh, but a bean. That's awesome. So on that note, I can't top that one. I'm going to go ahead